My name is Drew Heinmiller. I'm the product manager for the Vevo LAZR photoacoustic imaging system here at FUJIFILM VisualSonics. And I've got something really exciting to present today. Some brand new data that we've acquired using a brand new version of the software for the photoacoustic imaging system. I hope there are some existing customers out there right now who can benefit greatly from this and learn about the new software that's coming out. But I also hope there are some people out there who are not quite familiar with photoacoustics or even ultrasound, because I think there's a lot to be learned here as well. And I want to begin by sort of framing a bit of a challenge when it comes to molecular imaging and cancer research. I was reading this review recently. It's from the International Journal of Nanomedicine, looking at near-infrared fluorescent probes in cancer imaging. And it was a very nice review, a nice summary of a lot of the different fluorophores and nanoparticles and indeed multimodality and potentially therapeutic agents, theranostic agents, and this kind of thing. And it obviously revolved around optical imaging. And the image you can see up here on the left is one of the images that was presented. And this image is actually showing tumor-specific uptake of a fluorophore. And this dye, basically, when it's injected intravenously, it has a preferential leakage out into a tumor. Okay, and this is without any kind of targeting at all. And this is very nice. There are a lot of implications for this kind of thing. But there are some challenges as well with using this technique to learn about the biology, to learn about tumor biology. One of those questions is specific versus non-specific uptake. And what I mean by this is just an agent. It's a non-targeted agent that you're seeing here that's been injected IV, and it's taken up into the tumor. But I would call that non-specific tumor uptake. The mechanisms are still, not perfectly clear, the enhanced permeability and retention effect being one of them, but this is leaking out into the tumor by virtue of the tumor having different vasculature than other tissue and other organs. So it's tumor-specific, but how can we tell when we have a targeted agent, when we actually want to do molecular imaging and we want to see targeting there? How do we tell if it's targeted uptake or just non-specific uptake like you're seeing here? Another question is microdistribution. We can see, yes, that the tumor is highlighted in this image, but where in the tumor is this stuff accumulating? As we all know who work in cancer research, we talk about the tumor microenvironment because different parts of the tumor have greatly different biology, even in very small tumors. So we can't really learn much about microdistribution using this technique. Finally, real-time pharmacokinetics. How is the agent perfusing the tissue and how is it washing out of the tissue in real time? We can't really learn that using this technique. We can do, different time points and we can see where it ultimately ends up, but we can't really look at this in real time. That's to say nothing of other questions in tumor biology, such as just looking at things like tumor volumes. How did tumor volumes change with different therapies or drugs? How does blood flow and perfusion in these tumors affect the distribution of these kind of contrast agents? And what about tumor hypoxia? This is obviously a very important area for tumor or cancer research. So there's a big challenge here, a lot of questions that we're just unable to address using this optical technique. What I want to show you today is what I'm calling, the solution, and it all revolves around this image here. So this image here is sort of the end result of the work that we've done recently, and it addresses all of these points here. It addresses the specific versus non-specific uptake of a targeted versus non-targeted agent. It addresses microdistribution of that agent, which you can probably appreciate here if you look at the scale of this image. And it also addresses a real-time pharmacokinetics of this contrast agent. And of course, other aspects of the system, including the ultrasound, Doppler, and also the photoacoustics, which you're looking at in the top right image and the bottom right image in this slide, volumes, blood flow and perfusion, and hypoxia are inherent in the kind of imaging that we're doing. So we can help to answer those questions as well. And all in all, it ends up being a very, very powerful, photoacoustic and ultrasound imaging ends up being a very powerful technique for doing this kind of molecular imaging for cancer research. So what did we do? How did we get that image that I showed you in the first slide of the presentation? Well, first we have to use a contrast agent. So we can look at things like endogenous contrast, like hemoglobin, and get oxygen saturation, as I already mentioned. But to do molecular imaging, we need some kind of contrast agent. And the contrast agents that we're using are made by a company called Optimios. They've licensed the technology from Princeton. They've made a polymer photoacoustic dye-based nanoparticles, and they can be functionalized with small molecules and peptides and so on. Now, a key aspect of these particles is that they have differing absorption spectra. And what this means is that they essentially look different. They're different colors. And they can, Optimios can make many, many of these kinds of contrast agents, which really provides an advantage here, because now we can actually use more than one of these contrast agents at a time in the same animal. And this is a really, really key aspect here. So for this experiment, we worked together with Optimios, and we said, hey, you know, what do we want to do? We've worked together in the past. We've done a little bit of work with targeted contrast agents. What do we want to do in this experiment? And our goal was to image multiple photoacoustic contrast agents in vivo and assess the efficacy of molecular targeting. So how are we going to do that? Because, again, I pointed out in the last slide, because we have these differing absorption spectra, we can basically take agents of two different colors, okay? So Optimios made one agent. This is called the PAR 762 agent, which is one particular color. So it has, you know, one particular peak absorption. And then they functionalized it with either 5% RGD peptide or 25% RGD peptide. And we said, okay, you know, this is great. We can have different functionalization of this particle, but then we need a negative control. Here's the negative control. It's a different colored particle, essentially. The PAR 867 particle is our negative. So now we can really go after the goal of doing this targeting efficacy. So the hypothesis that we came up with is, well, you know, a higher level of this targeting. RGD, by the way, is a short peptide which sticks to integrins, which are overexpressed in many types of tumors. If we can, you know, this one's more functionalized with RGD, therefore, hopefully, you know, the targeting efficacy of this one is going to be higher than the targeting efficacy of the 5%. The negative control is really to get at that non-specific uptake and imaging the non-specific uptake and then being able to say, well, this is going to be our signal from non-specific uptake, this yellow particle essentially, and this is going to be our specific signal, this green signal, from our specific or targeted uptake of the particle. So first things first, what do we have to do? Well, to begin the experiment, Optimios sent us these contrast agents, and we want to characterize them first. And there's many different labs who are working on not just using these, but actually building these kind of contrast agents. So we decided, you know, let's help them out. Let's build a product here. So we got the Vevo Phantom Imaging Chamber. This is just a, you know, a acrylic phantom with these channels where we can put tubing, which we use, pardon me, to image the contrast agents in a very simple way. So it's easy to load, install them in the chamber. It's easy to load them. You only need small volumes of the contrast agent, so you're not wasting a whole bunch of contrast agent. So what we essentially do is put tubes in the phantom and then fill them up with the contrast agents. Then we just fill up that chamber with water, and then we image it with our system, with our regular probes. Okay, so this represents, you know, our two different types of contrast agents here. And this is the kind of image you get. I really should have 3 different tubes here because we were imaging 3 different contrast agents. So 5% PAR 762, 25% PAR 762, and then the yellow PAR 867. So we've provided this guide in the software where we can line up our tubes. You're now looking at cross sections of these 3 tubes. And then we have this acquisition setup. So you select the Spectral acquisition, and then you select the range. So we want to scan the entire range at 5 nanometer step sizes. And then we hit scan. So it's just a single button for acquisition, one click for the acquisition. And then you'll see that the changing signals here, and those changes represent the change of signal intensity with the change in wavelength. And what you end up with when this scan is done, you draw ROIs, and you generate these spectral curves. So I just want to point out the wavelength is on the x-axis here, and the signal intensity for each of these 3 ROIs is on the y-axis. So right away we can see the differing absorption spectra of these contrast agents that Optimios has made. So we've got absorption here at around, a peak absorption at around 770, 775 for the green agents, the PAR 762, and then the PAR 867 absorbs way out here at 875, 880. So these look different, and this is great because now we can separate the negative control here from our targeted particle here. The next step here is to actually save these spectra into the software. So we merely click up here, this dialog is brought up. The region, we can select the region, give it a name, and then give it a default color. Then we can even create groups and subgroups and divide these up into different things depending on how many of these agents you're testing. So what we're doing here is really saving these curves into the software so we can inform the software later on for both workflow and for spectral unmixing. Another nice feature here that's been added to the new software is if you go into Preferences and go into the PA Spectral Unmixing tab, you'll see all of the contrast agents that are saved currently in the software. You can display the curves to look at them. Not only that, but you can also display the curves that are built into the system, such as oxy and deoxyhemoglobin. So I can compare, this blue curve deoxyhemoglobin, red curve oxyhemoglobin. I can compare those curves to my contrast agent curves and see where their peaks are, where the values are, and where the best results are going to be for spectral unmixing. So this is sort of the total inventory of all the curves that are saved. Okay, let's go in vivo. So we've characterized our contrast agent. The next step is to go in vivo with it. Let's do an experiment. Here's the experimental design. We have 2 groups of animals. Each animal's, incidentally, just had 1 tumor on each hind limb. They're Lewis lung carcinomas. Maybe not a particularly interesting model, but that's what we have to work with here. And then we separated these animals into two groups, one 5% group, one 25% group. So the 5% group gets PAR 867 and PAR 762. So essentially, 5% RGD targeted. So we essentially just combine these in the same syringe and inject the combination of those 2 contrast agents at the same time into each animal. And then, of course, for the 25% group, we have the negative control, PAR 867, and then the 25% RGD targeted PAR 762s. And again, we combine those together and then do the injection in those 2 animals. We then scan before the injection, we scanned during the bolus, and we scanned 48 hours later. We quantified at each of those time points. So here are the curves that we've saved from those contrast agents. We know those, but we're going in vivo now, so we know that there are other things to contend with here, other absorbers, namely melanin, the pigment in skin will absorb it in the near-infrared range, and then oxy and deoxyhemoglobin. So now in the acquisition, and this is much different from the old version of the software, the acquisition is much different now. Instead of selecting the particular wavelengths and having to guess at which ones are going to be best to use, we can now just go into the Unmixing tab here, select the components that we've already saved in the software, and here they are here. Okay, so I've got 5 components I'm trying to unmix: the negative control, the targeted particle, oxy and deoxyhemoglobin, and melanin. And then the software will select which wavelengths are best used to do this imaging. We wanted to offer some flexibility, though. If you wanted to enter more wavelengths, indeed you can. So you can just manually enter the wavelengths that you want, and the software will add those into the wavelengths that it will use. So this is a multispectral imaging. We need to image at multi-wavelengths. And then you merely hit go and you're ready to go. So the next step is, okay, so now we know what kind of scan we're going to use. The software knows which components it's looking for, so it knows which wavelengths to use. But I want to image during the bolus. I want to image during that injection. So we've got an injector pump, which can be hooked up to the system. It can be hooked up to the image-guided injection arm. And then you can see here I've loaded a syringe, and this is all ready to go. In addition to this, we've integrated the software into our software, the ability to control the injector pump. So we can select the syringe, we can select the rate of injection, the volume of injection, and so on. And we can operate the pump essentially manually as well here by using this fast infuse and fast withdraw. So we have complete control over that pump now, and we can do that while the enclosure is closed. Our animal's hooked up to it, we're all ready to go, and we can perform the injection. And this is essentially what it looks like. So this is, an image, a little bit sped up image of the live acquisition. So we're imaging at several different wavelengths, remember, and that's why you see a fluctuation in the image there. And the two arrows that you see appear there are the beginning and end of the injection. And it's kind of hard to see in this image, but they're actually marked. Another software feature, they're actually marked on the actual cine loop. So the particular frame where the injection began is marked, and it's actually automatically annotated as well to tell you at the end here how much was injected, what was the rate of injection, and so on. So this is really, really nice functionality to be able to keep track right on the image itself of when certain events are occurring. This can be done completely manually, too. You just hit a button on the keyboard, and it'll mark the cine loop, and you can annotate it yourself. Now, what you're observing here as this goes, so starting here, you see some change, which is mostly due to oxy and deoxyhemoglobin, but as the injection happens, you see sort of an overall increase in signal at some of those wavelengths. After this acquisition is done, it's sort of difficult to appreciate here, but after this acquisition is done, we're brought into this panel here down at the bottom. This is the Spectral Unmixing panel. The components that we selected for the unmixing acquisition are already selected here. So we don't have to go back and select anything else. It already knows what components we're looking for. We just have to hit the process button, and that will spectrally unmix and multiplex our image. And that's what you're seeing right here. This is just a single frame after the injection of an unmixed multiplex. So what are we unmixing? We're unmixing oxyhemoglobin in red, deoxyhemoglobin in blue, our green PAR 762 particles at 5%, our PAR 867 particles, the yellow ones, and then melanin as well. And then these are all overlaid here. And if you want to appreciate this sort of more real-time aspect of this, this is, again, a sped-up version of the actual injection. So this is pre-injection, and you can see right away when that injection happens, you see this huge flood of green and a little bit of yellow signal coming in upon the injection. And this is the image that we want to see. Components are unmixed, and we're seeing essentially in real time how those components are going into that tumor, going into the circulation. Now I want to talk about quantification. So now that we have those images, how do we quantify those images, and how do we pull the data out of it? So I just showed you this acquisition. So after that, we simply draw a region of interest. So I've just drawn this region of interest here, and there's a whole bunch of different parameters we can look at here. It's going to measure the extent of each component, and because this is on the bolus here, it's going to measure the extent of each component over time. Now, I'll show you this here. All of the layers are displayed here, and we can select or deselect which layer we want to see. There's a whole bunch of really nice graphing features, which I actually don't have time to get into all of them, but a whole bunch of really nice graphing features which have been implemented in the new software. And one of them is to be able to sort of customize the lines that you're using here and markers, but also which agents you're actually looking at. We're looking on the time scale now. So remember, this is during the injection. So just before the injection, and then upon the injection, you see this huge increase in signal here and a small increase in signal here. Those signals have been unmixed. So we know that this signal, this huge increase in signal here is coming from those PAR 762, those green particles. and this signal is coming from those PAR 867 particles. And you say they wash in relatively quickly, and they stay in. They seem to plateau and stay in the circulation here. Now, here was something that was, and that's, again, the live image that we're looking at. Now, here's something interesting. This is the injection of, we are injecting in another animal, and this is from the second group. So this is from the 25% RGD group, and of course with the negative control. And now we can see a stark difference in the pharmacokinetics of these particles. This green particle shows, similarly to the 5% one, a huge increase, but it also shows a decrease over time way, way faster than the other one. And if you want to compare them side by side, here you go. So the 5% particles go in and they stay in. The 25% particles go in, and they're actually coming out of the circulation. They're washing out of the circulation quite quickly. So this is about, I don't know, maybe 14 minutes or so here. And I've actually resized them, so the time scale is the same. So over the course of 14 minutes, they're coming down quite low, whereas, again, the other particles, the PAR 867, the yellow particles, come in and they basically stay in, they plateau. So this is a really powerful way to look at this real-time pharmacokinetics. We can really see what's happening with these particles. The next question is, well, okay, they're coming out of the circulation, but where are they going? Why this difference? And can we see this difference somewhere else, in the anatomy of the animal? And we did this. We did a measurement. Now, this was actually post-mortem. We perfused the animals. We took out the livers, and we took a look at them. But this could just as easily be done in vivo as well. This is nanoparticle uptake in the liver. So now we're seeing that this is the green particle, the yellow particle. In the 5% group, there's far more, of the green particle in the liver than there is the yellow particle. But for the 25% one, the ratio is way different. So there's way, way, way more of the 25% RGD particles being soaked up by the liver than there are these yellow, the PAR 867 negative control particles. And then we can say, and I think with a reasonable amount of confidence, although this hasn't been fully validated, that, well, we've got 25% of that surface of that particle essentially has an RGD peptide on it, versus 5% on the other one, and the rest is polyethylene glycol. It's a molecule that basically shields the particle from the immune system. So here we have less exposure of the polyethylene glycol and more of this ligand. Therefore, the immune system is going to recognize this and pull it out of the circulation faster. So again, that's exactly what I strongly suspect you're seeing here. It's going in, we see a huge perfusion, but the immune system is recognizing this particle way more than it's recognizing this one, and therefore it's coming out of the circulation and indeed going into the liver. So really, really neat results here and very, very powerful technique. So now you can appreciate that if you're developing these particles, there's going to be a balance here between how much targeting you have and how long these things are going to stay in the circulation. Okay, I want to talk about microdistribution now. Let's get back to the tumor. Now, if you can appreciate the scale here, this is looking at 8 to 11 millimeters here. So there's about maybe 2.5 millimeter depth of this tumor. So it's a very small tumor. But of course, with high resolution, with high frequency, we're able to see it with very high resolution. So we can see, the skin overlying the tumor itself. There's a bone here which is causing this shadow, and then you see musculature of the hind limb underneath that. Now, because we have our unmixing, we can unmix for blood. So that's what you're looking at here, red being oxygenated hemoglobin and blue being deoxygenated hemoglobin. And you can see, again, there are massive differences, even in a tiny tumor like this, in the distribution of oxy versus deoxyhemoglobin, and indeed just blood itself. So another parameter we can look at is just total hemoglobin. Here's a branch of the femoral artery that you're seeing down here, but you can see here that there is really non-uniform vasculature within that tumor. Now let's add the signal from the particles. By the way, this is 48 hours later, so we don't really expect any of these particles to be in the circulation at this point. They should all have been cleared from the circulation. So this is deposition of these particles in different tissues and different areas. So here's our negative control. So indeed, the negative control particle does come out of the circulation into different tissues. It comes out quite a bit in the skin, but you can also see that it does come out into the tumor as well. And this is this non-specific uptake that I've been talking about. We can see, you often get this, what's called this ring effect, where around the rim of the tumor you do get non-specific leakage of these kind of agents into the tumor tissue. And that's what we're seeing here with this particle. Now let's take a look at our targeted particle, the green particle. Now you can appreciate why having this high resolution, being able to see the micro distribution is so important. If we were just looking at this from a surface view without the resolution that we're getting here, it would be impossible to distinguish these two signals from each other. Maybe relative amounts, but so difficult to be able to say, okay, well, there's no non-targeted particle here, there's no non-specific uptake here, but oh look, there's plenty of signal from the targeted particle coming out here. Now, I want to spend a little bit of time on this because I think this is very significant, being able to see with this high resolution microdistribution. We're really talking about microenvironment of the tumor now. What is the microenvironment of this tumor? Why, other questions arise. Why are particles depositing, here and not here, for example? There's a lot of questions that come up here, and this image is I think, a very powerful image and a powerful demonstration of this technique. Now, let's get back to the quantification. What do we do with this data once we have it? Well, a couple of things we can do. There's one thing we can do, really simple thing we can do in the software, and that is simply to do a very simple calculation, in this case, just a subtraction. So let's take essentially the green signal, and then let's subtract out the yellow signal. We know the yellow signal is non-specific uptake. Let's subtract that signal from the green signal. And that's exactly what we're doing here in this panel. So you can just do it very simply in the software. And sure enough, you see now the green signal that you're seeing is actually the green signal that's left over after you've subtracted out the signal from the non-specific uptake. So just plain old doing the subtraction is one way to do this analysis, and then we can quantify this as well, which I'll get to. So there's one really, really simple way of doing it. But I thought there's got to be a different way. We know this stuff is being taken up in the skin. We know, in fact, both types of particles are being taken up in the skin. It would be ideal if it was just one and not the other, but that's not the case. So let's take a look at essentially different organs. Let's take a look at the skin versus the interior, and let's do a ratiometric analysis. How much of each particle is being taken up in the skin versus the tumor? And we can do this because we have the high resolution, because we have the ultrasound, the anatomy. We can really do this with very, very high precision. So we're just drawing ROIs, regions of interest, that encompass the skin versus the tumor interior. And of course, we can do this in 3D as well. So we can draw these 3D regions to have, a 3D volume containing the skin and a 3D volume containing the tumor interior as well. And here's what we end up with. So let's take a look at this one first. This is the 5% RGD divided by the 867. So it's a ratio of the one to the other in the tumor versus the skin. So let's look in the tumor first. So a ratio above 1 is obviously going to show you that there's more of this 5% particle than this 867 particle in the tumor. So that's great. We were expecting this targeted particle to go to the tumor more than the non-targeted. However, there's also more in the skin. But if you look at the ratio of these two, proportionally, there's actually more of the targeted particle in the tumor than the skin. Okay? I've made the scales equivalent here. So in this case, remember our biodistribution. Remember that this 25% particle comes out of the circulation so much faster than this 5%. So there's way less of it. Essentially, there's actually more non-targeted particle in the tumor and the skin than there is the targeted particle. However, again, if you look at the proportion of the particle one to the other, you can see that in the tumor interior, proportionally, there's actually more of this targeted particle in the tumor than there is non-targeted if you compare it to the skin. And this is the figure that kind of says that all. Let's now look at the whole goal of our study, which was targeting efficacy of the 5% versus the 25% RGD. And we can see that, again, even though we have overall more of this 5% in the tumor, if you actually look at the ratios between these two, the proportion between these two, you can actually see that proportionally there's better targeting efficacy of the 25% RGD loaded particles versus the 5% RGD loaded particles. Again, not really that surprising. We would actually hope that that's true, but now we're able to look at the pharmacokinetics and the microdistribution and put those two pieces together and actually learn something about targeting efficacy. We can learn about targeting efficacy versus just non-specific uptake. This is a simple graph. It's only 4 animals. It's early data, but it's very, very promising. Really nice, powerful technique here. So I want to come back to this again. We've answered these questions here, specific versus non-specific uptake, microdistribution, and real-time pharmacokinetics. We've answered these with this image here and this data here that we've taken a look at. What we haven't done, but we could do, is take a look at what's the influence on the tumor volume on these parameters. What's the influence of the blood flow and the perfusion on these parameters? What's the influence of hypoxia on these different things? How do these factors influence the uptake, the specific versus non-specific, the regional microdistribution of these agents? How do these factors fit in? And frankly, I just didn't have time to look at all these things. But the data is there. We have the volumes. We have the blood flow and perfusion. We have hypoxia measurements. Now we can start to relate all of those to the data we're looking at. And now we can say a huge amount more than about tumor biology, about molecular processes that are going on than we ever could with this image here. I just want to end off by talking about some applications. Really quickly, just a few slides here. What I want to start with, it actually doesn't involve photoacoustics, but it's very exciting. This paper just came out, the Journal of the National Cancer Institute. It just came out. It's from Bob Kerbel and Stuart Foster's group and Andrew Reynolds at Sunnybrook Research Institute. And it's a really groundbreaking paper, basically showing that tumors switch from neoangiogenesis to normal vessel co-option. So they take over normal vessels when they're being treated with an anti-angiogenic drug. So essentially they're able to switch from this phenotype of neoangiogenesis to this phenotype of vessel co-option, and that's how drug resistance is expressed. Really groundbreaking work. This is huge. This is the first time it's been shown. And I do want to point out, of course, that contrast-enhanced ultrasound was used to assess the perfusion. Okay, so contrast-enhanced, we have these small microbubbles. We inject them in, and we get a very strong ultrasound signal from these microbubbles, and we can assess the perfusion because the microbubbles stay within the vasculature. They don't leak out, as opposed to the nanoparticles. The microbubbles are much bigger. They don't leak out of the vasculature, and we can assess the perfusion right down to the capillary level in the control animal versus the anti-angiogenic treated animal. And what this group did is they actually looked at this functional assessment using microbubbles and compared it with histology, and they really found some nice correlations between, for example, this very, very highly perfused rim, which before was suspected to be just neoangiogenesis, is actually a result of vessel co-option. Look up the paper, read it. It's wonderful. It's groundbreaking. It's really, really great work by this group. Another application I want to point out is that there are other manufacturers of particles for photoacoustic imaging, and one such company is Nanohybrids. They make gold particles for photoacoustic imaging, and others as well. They are also tunable, so we can also tune them, basically make them, absorb at different wavelengths, make them different colors, if you will. And they're also silica coated, so they're much, much more stable than other particles of that kind, which are not coated. And of course, these ones, similarly to the ones from Optimios, can be conjugated and functionalized. So this kind of molecular imaging can be done with these as well. You can visit them here at nanohybrids.net. But I want to point out some work that was done using their materials, which was published in Cancer Research, I believe last year, from Stanislav Emelianov's group, where they essentially took these particles and functionalized them so that the targeted particles will go inside the cells and will actually have a different photoacoustic signal than particles which are just floating around outside of cells. So these are EGFR targeted, so they're targeted to these cancer cells, and they developed this metastasis model where they had a primary tumor in the tongue. It metastasizes down the lymphatics into the lymph nodes in the neck, the cervical lymph nodes here. And then they injected their particles into the tumor. The particles then drain to this sentinel lymph node here, and then those particles essentially get sucked up or taken up by metastatic cells there. And in fact, metastatic cells which are not yet visible even with high-frequency ultrasound. So we can't see them yet with ultrasound, and yet we can see photoacoustic signal that's coming from particles that have been taken up by those cells. So we can therefore identify metastatic cells even before we can see it with just the regular ultrasound. So this is a great example of true molecular imaging for detection of metastasis using this material from nanohybrids. Another paper I want to point out, it was published again last year in Theranostics. This is by another group here in Toronto. They were using a head and neck tumor model, so this is a really clinically relevant model. They were using it in rabbits, I believe. And they were doing this multimodality imaging or multimodal imaging, and I've been to a few of these molecular imaging conferences over the past few years, and that seems to be a trend towards being able to see a certain contrast agent, not just with one modality, but with many. So for example, looking at something with fluorescence, which is practical for something superficial like head and neck, looking at it with fluorescence as well as photoacoustics, and then on top of that, having these particles functionalized so that they can be used for photothermal therapy and other types of therapy. Also, being used to detect metastasis. So again, I'm not going to go into more detail than that, but please check these papers out. Really interesting, really amazing potential for photoacoustic and frankly other imaging here for cancer research.